Everyone, and welcome to Module 9. This is already week 9 of the course. Few more weeks to go. I guess you have roughly 4 or 5 weeks, including the break that is going to come sometime in November. So, in this course so far, we have been exploring more and more about bioinformatics. It is introduction to computation methods. We are trying to refresh you with all the concepts, I would say 101 bioinformatics concepts, all of these core concepts that are needed, sort of some of the material in this lecture and the coming lecture. Some of the ideas would seem, oh, I've watched this before, okay, so probably from a previous lecture. We have discussed a similar idea and so on and so forth. So we covered the foundation in a good in-depth. Still, we need to get your attention into some important concepts. Hopefully by now you are also able to recognize some tools. You had these in-class activities. I know many had some issues. Don't be frustrated. This is natural. I still remember at my beginning, I was dealing with this terminal and I had to figure out running a program. It was just the beginning, and then it just sometimes it's not that you are not able to understand. You basically can explain many things much better than I do. The idea is it is new, totally new for us, especially those first time for them to see terminal, compute cluster, and discovery, and so on and so forth. So I still remember this story where I had to call an expert from what's called the scientific computing team. This was an interesting team as part of the data center and HPC services and my PhD institute. Anyway, the guy came, and a good friend, and I don't know, like I spent hours, and then he said, "Show me the error." Showed the error message. Showed the error message. Said, "Okay, do this and that." And got fixed. And then I still remember what I told him. I told him, "Okay, I mean, you are doing it very easy. It took me hours. You are much professional and expert." He told me the response that I get from that expert was, "You will get it. You will be there. Just be patient." And indeed, I still remember this story because after a few years, I was there, I was doing it, I was helping others. So you will be the same thing if you have, the dedication and interest. So you are dealing with the terminal, you are running already some bioinformatics software and tools. Today, we're going to focus into genome sequencing, okay? We covered some ideas of genome sequencing, so we're going to try to touch on some of the points, again, that may seem kind of review and refresh for us. At the same time, we may get attention to important ideas, as I've mentioned. So first, I want to, make sure that I'm hoping everything is working fine throughout this course. I know it makes you feel a bit busy. There is a weekly task, but so far so far I'm hoping that all of you are building gold experience. I can tell there are some differences in terms of how I see responsive studies. This course builds really on your interest. There is this hope that you are not here in a master's degree in bioinformatics unless you are interested. It's not more on exams, it's more on reading, understanding, having discussions. That's why right from the beginning and until now, it's not this discussion anymore that we have in undergraduate courses. Yes, we have lots of tasks, weekly tasks, loaded tasks, but the point is not meant to be, I'm finding a way to cut marks from you. No, the point is we want you to be pursuing a path as a professional bioinformatician. So reading is always important, especially in these new emerging fields. So I hope you have been working good in the tasks, trying your best to read and understand. If you have been doing so, and you think you are spending more hours in this course, that is great. That's mean you are achieving the benefit. Okay, maybe somebody else would do it throughout the course. I don't know, with pass, but then without spending that much time, then the needed and necessary outcome is not was not achieved. I can tell you from experience, whatever we are discussing here is very foundational to any bioinformatics position. Today we're gonna, so again, as part of the check-in here, what I'd like to ask you about your course, about your project progress. So hopefully in the next coming office hour, I can, discuss this more with you. But I'm having good hopes, okay, that you are working on your projects. And remember, you have to produce some results. Doesn't need to be too complicated. You need to focus, okay? By now, you should have read your paper a few times as a team, had a meeting to discuss, found some GitHub codes, run that, got some output, and you are on your way to find some part that you can reproduce. And then you are reporting in your presentations if you find any discrepancies as compared to what they have reported, or you find some, issues in their own protocol, or whatever they reported, or the challenges you are facing, and some of the missing information that were not there. So otherwise, in today's lecture, we're gonna look into genome sequencing. Oh, let me actually, as part check-in, ask this question as the first treasure hunt question, which is how are your team, how your team is doing with regard to a project. Okay, just tell me if there is good progress. I've just told you what I'm hoping, at what stage you are. So tell me if you are there, like, have done the reading, decided on something, started running some code, and you're getting there. So please share with me your reflection or whatever you have been going through with this part. Okay, so today we're more into genome sequencing. We want to understand again or refresh with genome sequencing. We looked into sequencing technologies. We looked into genome sequencing, the jigsaw puzzle. But again, we're going to review some terms here or some ideas and go a little bit more into details with some other ideas, and then discuss whole genome sequencing and whole exome sequencing, and finish up with ChIP sequencing or ChIP-Seq, okay? And then now, and also next lecture, we're gonna dive more into RNA-seq analysis and RNA-seq methods. There are different and many types of RNA-seq methods, and also look into transcriptome and start linking things together. So hopefully also a little bit more about RNA-seq data analysis. So in terms of learning objectives, hopefully you will be able to fully describe what is genome, and there are certain terms when it comes to genome sequencing that we need to make sure we understand, such as, whatever is gene, chromosome, genome, contig, scaffold, and so on and so forth. What is whole genome sequencing? What is whole exome sequencing, whole genome sequencing, and ChIP sequencing, ChIP-Seq sequencing, and so on and so forth. There are two strategies I will discuss as part of the genome sequencing, one that refer to as hierarchical clone by clone, and shotgun. Genome sequencing, and why genome sequencing? Genome sequencing, as compared to what we discussed before in terms of, looking into gene expression, right? For gene expression analysis, we are doing microarrays. We looked into RNA-seq as well to try to read cDNA eventually that is coming and reflecting on the quantification, which is the extracted mRNA. So certainly when you start speaking about genome, we are looking into extracting the purification and extraction of DNA. So in this case, it is generic in terms we are trying to go to the reference book, that full library of genome, as compared to focus on specific disease conditions where we would look into mRNA study and analysis and so forth. So here I would ask the second also treasure hunt question: what is the difference between RNA-seq used for genome sequencing and RNA-seq that would be used for gene expression? So tell me what that brings to your own brain, and in terms of memories from this course, or some ideas that you still remember, hopefully, and so on. So share your insights. So genome sequencing, it could be the case that sometimes we are interested in building a genome, even if we discussed this before, right? We may have a genome, that's the human genome, that's the human project. But in certain continent or country, we may have a different version of genome. So why not to take advantage of that existing reference human genome and then come up with a genome for that is unique for that land or that population and so on and so forth. That could tell us something about some variants that we're looking at, and try hopefully to highlight certain factors of differences. Another thing, sometimes we could end up working on a project that is part of the environment. I still remember this project. We're looking into environment species, and one of the species was not interesting enough to the big institutes. Remember, we were talking about EBI, NCBI, and so on and so forth. So these institutes did not actually, and even some labs in the world, maybe done some preliminary efforts, but still that specific species did not have the genome out there. So also genome assembly, sequencing and genome assembly is very valuable sometimes for specific projects because of specific needs. So genome sequencing. Genome sequencing is what we want to look into here. And of course, there's this question, which is, what is a genome? And I'm going to hide the slide and ask you, in few words, as a treasure hunt question, what is genome for you? Okay, what is genome for you? We have described certainly genome before, but after you progress with this course, yes, you can say genome is a set of nucleotides and so on and so forth. But what is it for you? Okay, like for you, what does it have there? Okay, no question that we know that it's a complete set of DNA is what we call genome. DNA is our genetic code and information. So it's the molecule of life, has everything. And DNA, is made up of these nucleotides arranged in these set of chromosomes that are wrapped around histones and so on and so forth. So we have this compact structure. But then we don't have one chromosome. That these chunks of DNA, we end up with 23 pairs as human of chromosomes. If we have a way to read all of that information, and I repeat, all of that information, this is what we call genome sequencing. Okay, genome information. That is the genome that we're looking at. So specifically, the genome is the entire set of DNA. Or perhaps if we would refer to a virus, we'd say the RNA. That is, it includes all of the genetic information of an organism. Now, DNA is having coding regions, okay? Sorry, the genome eventually is going to contain both, okay? It's ordered as set of chromosomes, okay? And in each specific site or part here, we will have some coding and non-coding regions. Some coding regions that would refer to genes, and some non-coding regions. So we have the human. We know that the human genome consists of about roughly 3 billion base pairs of DNA that are 3 billion that are split across 23 pairs of chromosomes. Okay, pairs of chromosomes. The chromosome itself is a long and continuous piece of DNA. It has many genes as well as regulatory information and other non-coding regions. In general, many organisms have multiple chromosomes. Now, each chromosome has many genes and also non-coding sequencing regions, as we see here in our picture. When it comes to the gene, it is this specific, the gene here, it is this specific sequence of nucleotides in DNA or RNA. How do we see this part? It is the functional unit of inheritance, eventually controlling the expression of some maybe one or more traits. So this is one of the very important, I would say, secret or hidden instructions. The reason I say hidden instructions, for our biological system, it might be clear, but for us, when you see it looks like ACGT, some sequence of nucleotides. But remember, this part is the one that is given, being read, and responsible for generating eventually mRNA and then proteins and so on and so forth. They have specific functions, okay, and they are located at specific positions. Okay, for example, see if you would look CFTR gene in an online database from NCBI, you remember the module about databases. CFTR gene in humans, well, let me ask you this. I'm going to give you a moment and let's have this as the treasure hunt question. Can you check in which chromosome we have this gene? So let's do this exercise. I'm going to give you, like, pause a little bit, and then so you can have your time. Of course, it's going to take longer to check, but enjoy the exercise. You know, if you want to write any number, you don't want to do the exercise, it's just fine. As I said, always there are no right or wrong answers for the treasure hunt questions. So can you find out where is CFTR gene in humans? Okay, specifically what chromosome it is located in, and what is it responsible for? Okay. Let me give you a few seconds here. So CFTR gene is, and again, share with me what database did you look that information in, is located in chromosome 7. And it's responsible for the production of a protein called the cystic fibrosis transmembrane conductance regulatory. And this is that if there are any mutations in this gene, is going to cause cystic fibrosis. So in summary, genome is the all genetic material. Chromosome is one piece of tightly packed DNA, that is containing many genes, and gene is a specific sequence of the DNA. And I like this example. If you want to visualize this, you can think of genome as library. Okay, this is the full library. Then the chromosomes are like the shelves. Okay, and you can say that the genes are like the books on these shelves. Books. I would say the group of genes. Okay, all these genes are going to be kind of the books. And the story inside each book would tell specific instruction on with regard to protein, making protein, and so on and so forth. So genome sequencing is certainly this discussion that we had before, right? The jigsaw puzzle, where we are not able to take the whole genome in one shot and read the whole DNA in one shot. So we end up breaking it down in different strategies. We're going to highlight today maybe 2 strategies, but we're going to end up with some reads. And this is kind of the newspaper example, right? Put a boom there and then some dynamites, whatever, boom, and then you end up with these repeated. It's the same newspaper. We have 1 copy, 2, 3, 4, and many copies. Boom, there you end up with puzzles. This is how many would like to say it is. Okay, and so on and so forth. Okay, so then you start aligning these pieces together, try to get the full genome. Well, boom here is what we can refer to as shotgun, as well as the technology is telling me I have to break down the full genome into tiny pieces or small pieces. And then, I need to start getting the whole message. I need to start getting the full DNA information. So genome sequencing is this process of finding the complete DNA sequence of an organism's genome. This is a process. It is a demanding process. It may require lots of computational resources to find the right order of nucleotides, specifically adenine, thymine, cytosine, guanine. Okay, all of these. What you find here is, there is some issue here and this, so that is starting string. So the first one, this was replaced. Okay, this should be I, and the second case is R is missing from the string. In any case, this is just an analogy, an idea to say we end up with some sophistications. Okay, some sophistications where kind of we are missing some word here, kind of we are having some replacement here. And also we mentioned this before, sometimes we don't know the source of this. It could be an error because of the protocol of the machine, of the algorithm, or it could refer to something unique about this sample. Genome sequencing is a process where certain steps are followed, starting with sample collection, library preparation, sequencing, assembly, and then annotation. Now, this has lots of applications in different domains, as you see here. For example, in medical research, genome sequencing is crucial in identifying genetic variants, those associated with diseases. Remember from a previous lecture, I've discussed specifically when it comes to diseases like cancer, for example, and when we want to do the genetic testing for one of the patients where we screen the genes they have, we want to see the mutations, right? The specific mutations based on the libraries and databases we have. Remember these variance databases that we looked into before. So anyway, we take that gene sequence for that patient, but usually we look into maybe roughly for breast cancer, for example, I guess 18 genes or so to try to see if they have breast cancer, the potential for ovary cancers, and so on and so forth. Ovarian cancer, or yeah, I think that's the name, ovarian cancer. So they are related somehow. So we end up screening 18 genes. But if we do this genome sequencing, imagine for the whole genome, this can be give us a better diagnostic treatments and understanding of many disease-related mechanisms, and that specific person or organism or sample. Comparing genomes across different species can give scientists the chance to understand things about the genetic basis of adaptation. Sequencing the genomes of bacteria, okay? Sequencing the genome of bacteria, viruses, and other microbes can help us in understanding their biology and the resistance mechanisms. Who knows, in the future, also individual genome sequencing, if we're able to easily sequence the full genomes of individuals, this may allow us for finding tailored treatments. And there are lots of other applications, like even in agriculture and crops and livestock, the genome sequencing can help us identify genes associated with some traits. That's where some are interested in developing, the breeding strategies, eventually enhancing the meat, flesh quality. I referred to some story before, research story before in this course, from a friend who was doing research in China on fish samples. Now, the part of this process that we're looking at here, again, there are lots of terms here: functional genomics, looking into knowing the functions and specific function parts; epigenomics, the what's behind those functions, trying to release some of the parts or the roles of certain areas behind the major functions in the DNA. Comparative genomics, as I mentioned, looking into microbial information and other different, metagenomics. When we have samples of mixed nature, and we try to all do these different analysis and different genomes or different DNA samples all together, genomic medicine, as I mentioned. Yet there are some complexities. Okay, some genomes are highly repetitive or complex, so this increased complexity. Although the genome sequencing has dropped significantly, it can still be a limiting factor depending on the nature of the project and study. And the vast amount of data produced by genome sequencing usually requires powerful computational resources and expertise, would require expert bioinformaticians like you to go and do the work. So here is one first strategy for doing what's called clone by clone sequencing. This is also known as hierarchical sequencing. This is used to sequence large genomes. So it usually involves breaking down, or it involves, I'm going to omit the word usually because this is clone by clone, it involves breaking down the genome. In fact, all genome sequencing methods that we're going to discuss require we have to break down the genome into manageable pieces, and that step is what we refer to as the fragmentation step. Of course, before that, the DNA sample would have been obtained from the organism of interest, so that is sample collection. Okay, now we are into the step which is known as library preparation. So the DNA is often fragmented into smaller pieces, and specific adapters are attached to facilitate sequencing and so on and so forth. Now, sequencing can be done in different ways. As we discussed before, there have been several sequencing methods over the past few decades, starting from Sanger sequencing, which was the first method developed and still used for specific purposes, to next generation sequencing that can process large amount of DNA in parallel, like Illumina, until we went to third generation sequencing, like Oxford nanopore sequencing. So once we have and we are able to use some of these technologies to actually sequence the genome, we will end up with what? With reads, okay, with reads that we need to assemble together to solve the puzzle. And finally, as a general step, is we want to do annotation. Once we assemble and we have our constructed genome, okay, what do we do with it? We have to say where is some variants, some genes, where are the genes, the locations of these genes, and so on and so forth. So now, specifically, when we look at what's called clone by clone sequencing, as we see here, there are specific steps, okay? Breaking down the genome into, again, manageable pieces. So the entire genome is first broken into larger fragments in the beginning, typically ranging from 100 to 300 kilobase pairs, kilobases or kilobases in size. Okay, then these fragments are in a size which is suitable for cloning into bacterial vectors, like bacterial artificial chromosomes. This is something we covered before. So these are taken, and then this is kind of the genomic DNA. Well, we can think of this as the pack, and then that part of fragment is here, or in an opposite way, this is the fragment and this is the pack here. In any way, the idea is we have the vector here, and then we have our fragment that we attach both into this one. And why we are doing this is certainly a treasure hunt question, which is, again, why are we doing this insertion into bacterial artificial chromosomes or yeast artificial chromosome? I want you to think about it, okay, and share with me your answer. Very important that, this is something that we are reviewing here. So once insertion is done in DNA, which is going to do a specific important role, basically cloning, okay, that is cloning. And this is part of, we are in the step of creating a library. Eventually, we are doing this insertion here, doing the cloning, fragmenting again, and this kind of process is repeated until we are sure that we have the library. That is what's called a genomic library. A genomic library is a collection of DNA fragments, okay? That is going to represent the entirety of an organism's genome. So that is when we move ahead and start clone by clone sequence. And why it's called clone by clone sequence specifically. Okay, the idea here is clone by clone sequencing. It involves creating a hierarchical library of the genome. Okay, that is sequencing the fragments in a defined order. Sequencing the fragments that we had in some defined order. Okay, and so there are some tags that will be attached defining the orders for those fragments, and then assembling the sequencing to generate the full genomic sequence. That is what we can achieve through this process. Okay, so in other words, sorry, as we had the library created, we want to move into, into ordering. We need to realize that the fragments in the library are ordered. Or, in other words, to make it easier to understand, they are mapped these fragments are mapped, so are mapped to relative to their position in the genome. So basically, probably this is coming from here, this is from here, and this is from here. This would mean that this is going to be sequenced first, this is going to be sequenced second, and this is going to be sequenced third, and so on and so forth. So this order style, because we took this into consideration that at the beginning, is what makes this called clone by clone sequencing, okay? And until we recognize the full alignment and we find the remaining sequence. As compared to what is called shotgun sequencing, that this is another way to do it, which is basically random. Shotgun is you shoot, and then shotgun is yeah, I would say that you are destroying something in a random fashion. So you end up with random reads, okay? You end up with random reads. This is just an example. So what we end up as part of sequencing or sequences, the reads that we have. Usually these reads, I mean, this is a very, very nice example of what is there in real life, because easily we can overlap these, okay? The second, this one with this one, and we are still able to cover the original. And maybe we join this with this, which means we have two reads, and then we join them together. That is alignment, okay? These are easy to align, very straightforward, but in real life cases, this is not what we see. It's very sophisticated, it's very complicated. We even looked into some of the ideas that before are used in genome assembly algorithms, right? Like, kind of the, specifically an alignment, assembly can get to be very sophisticated, especially if we don't have a reference genome. You end up running some tools or working with generic or working with algorithms that are known to be intractable, meaning they are not, they take lots of computational resources and yet might not guarantee the best solution. Okay, so when it comes to shotgun sequencing, we are looking at a method that is used to sequence long strands also of DNA, such as whole genomes. That is what we are interested with. Instead of sequencing a genome piece by piece, that is the clone by a clone. There it was piece by piece. Shotgun is going to take a randomized approach, okay? It's going to take a randomized approach, and again, the fragmentation is the first step where there will be a difference as compared to the clone by clone. The DNA is randomly broken down into numerous small fragments, okay, into numerous small fragments. Okay, and so the size of these fragments can vary, but generally they are much smaller than those used in clone by clone, okay? There is then cloning. Typically it's similar. So these small fragments are then cloned into a bacterial vector, typically a plasmid, and this, again, this is what's gonna allow it to be amplified in bacteria, producing enough material for sequencing. And this is an answer for one previous question, but I hope you have answered it, okay? And then we go to sequencing. Each small fragment is sequenced then, okay? Each small fragment is, talking about this step, is sequenced then, and then we are gonna end up with assembly. Okay, this is where we had, yup, I think, let me see here. Yup. So we are constructing a large insert library from the genomic DNA. Very nice. Determine path. Okay. The shotgun. And then we have the sequence assembly and so on and so forth. Okay. Genomic sequence. So again, what we are looking into Here is for assembly. Once we are done, so let me summarize this. We are looking at fragmentation, okay, as one general step, then cloning. Okay, this has nothing to do clone by clone, what we meant there by that other approach. Cloning always takes part in the bacterial vectors. There is then sequencing, okay, and again sequencing where each small fragment is sequenced, such as using again single methods or next generation sequence and so on and so forth. To have, you know, we're looking to eventually have overlapped regions where we can use and call assembly software or algorithms to try to assemble the short DNA sequences based on their overlaps. Okay. Now, so this is what we see here. And part A depicts a whole genome shotgun procedure, starting by fragmenting the genome into pieces, and then the other part on the right shows the hierarchical approach utilizing a larger kind of library. So we can have up to 200. The first one can be starting by fragmenting the genome into pieces that are larger than 10 kilo bases. Okay, here we're looking at 200 kilo bases. Okay, so depending on the approach, we can start with the longer one, but then we do a hierarchy. And so there are these variants, okay, break it down a little bit and then farther and so on and so forth. So that allows us to do some hierarchy if we are tracking that window also was sort of what we refer to as a clone by clone. And again, when I say clone by clone, a different way to think about it is that we are, or when I say hierarchical, that is defined in order, okay? That is, fragments are defined in order, so we know the order of the fragments based on that. So also in whole genome, this is now, we move from shotgun sequencing to what's called whole genome shotgun sequencing, okay? Now, this is a specific application of shotgun sequencing because it targets an organism's entire genome. So the idea and the goal here is to get the full genome without targeting specific regions or relying heavily on some physical map of the genome. In other words, previously we're assuming maybe, you know, there are different applications. Shotgun sequencing, you might be interested in specific area. But in this case, we are looking into having the full genome, okay? And again, this is going to go through fragmentation, cloning. And let me remind you something also about cloning. In next generation sequencing technology, this step can be often bypassed, okay? And my question to you as a treasure hunt question, can you explain? Okay, so I'm not going to explain it further, but I'm going to repeat the statement. The cloning, which means you know, taking the fragments, inserting them into the plasmid vector and grow them in bacteria to get some enough DNA for sequencing later, okay? This is the general overview of cloning here. However, next generation sequencing, can be often bypassed. Actually, we explained all of this before, so I just want you to think about it and tell me what do I mean by that it can be skipped, okay, or bypassed. Specifically refer to the name of one of the genome sequencing or next generation sequencing used for that and why it doesn't need cloning here. And then we move to the sequencing part to read these fragments and finally with assembly. Now, before I just illustrate what do we have here, when it comes to assembly, and something we're going to also see later on. There is what's called de novo assembly. This is one type, de novo assembly. This means there is no reference genome. Okay, this means that we don't have a reference genome. It's not available, which means the assembly is done from scratch, and the only thing we can depend on is the overlaps between sequences. That is de novo assembly. Another type is what's called reference guided assembly. That is reference guided assembly. So if we have a closely related reference genome, then it can be used to guide the assembly and makes our life maybe a little bit easier. Okay, so keep this in mind. And again, the example of having the human genome and so on and so forth. Okay, so genomic DNA. So the whole genome, when we're having the genomic DNA, we want to achieve whole genome shotgun sequencing. So we can have sequencing of single end or paired end fragments of the whole genome, DNA shotgun library. I want to ask you here what single end and paired end means for you as another treasure hunt question. Something we discussed before. So what does single end and paired end mean for you? Okay. Okay. So sequencing of single end or paired end fragment of whole genome DNA shotgun library is what we see here. They are made with the average range of 200 to 800 base pairs. Okay, that is the length of these fragments. And again, it depends on the technology. So usually we have this, and it goes for Roche 4454 systems or Illumina Silixa systems. So in general, producing a total DNA sequence amount of 15 to 20 times and 60 times coverage and depth of genome depends on the system. Okay, so that's why if we always, whenever we find a name of technology, we need to know what they give us when they are gonna be used, and this is gonna achieve for us context. So we have short fragments that we can align or read, and so we can assemble, and so we get the context, which I'm going to illustrate the definition of context shortly. Okay. Now, when also whole genome shotgun made paired end sequencing for scaffold construction. So scaffold construction, this is when we are dealing with 2 to 40 kilo bases, okay, of random DNA. Then we are meeting both sides to try to establish the and construct the scaffold. Okay, and so on and so forth. So depending on the goal, we might be looking to construct the contig or the scaffold. Now, scaffold is larger than the contig, which is larger than the read. So genome sequencing in a nutshell, as I talked about before, library preparation, some pre-processing. Let me change the color here. Okay. Pre-processing, alignment, assembly, and variant calling. Okay. And specifically here, what we are looking at when we say variant calling, and again, it's the process of identifying differences or variants in the sequence genome when compared to what? Of course, a reference genome. That's when we start seeing something of interest there. Annotation is very important step. Okay, you see this part here. This is a very important step. Okay, this is a very important step, the gene annotation, because library preparation, alignment and data analysis, because we have some reference genome, we assemble the sequences, we start looking into the variants. Now, what about where are the genes? Okay, gene annotation is this process of adding the biological information to the sequences. What does this sequence mean? For example, identifying the locations of the genes or other functional elements. Whatever we do in bioinformatics, by the end, we want to think and be able to map this to some functional annotation, right? Think about enrichment analysis we looked into before, or gene ontologies, and so on and so forth. The goal is always eventually to try to come up with conclusions and see what this whole thing means. Also, one of the primary tasks in annotation is what's called gene prediction. So in this case, the genome is scanned for sequences that likely encode proteins, for example. So we may compare this based on databases to known gene patterns, or we do some comparative genomics, or try to find some evidences, and so on and so forth. Okay. So in a broader picture, once the DNA is sequenced and assembled, either we do this de novo or using a reference. Variant calling identifies the difference between the sequenced genome and the reference, and annotation provides insights into the biological significance of the sequenced genome. And both steps are essential for extracting meaningful, okay, both of the into meaningful what? Meaningful what? Biological information from raw sequence data. What is ACGT for us by the end of the day? We still need to recognize this information. So whole genome sequencing, and here you find some interesting insights about, or numbers about, genome sizes. This is going to raise some interesting discussions. All right, so wow. So you can look into here and you can, query more about certain information, like where viruses stand up here, bacteria, general, small fungi. And then until you get to mammals here, humans here are around 3 gigabases, 3 gigabase pairs. But when it gets interesting is when you see protists, okay? Specifically things like amoeba. Amoeba, the genome of the amoeba is 100 times larger than the human, okay? And sometimes an amoeba could have it more, okay? So we're talking about almost many billions of base, hundreds of billions of base pairs. Okay, let me check this quickly. Genome size to give you the for amoeba. Okay, this is going to be how many base pairs here. So we will have around 670 billion base pairs. Huge. 600. Human is having roughly 3 billion base pairs. So this is not 100 times even. This is 200 times. But amoeba is only single cell. So this is like a dilemma question. How come amoeba, which is single cell, very simple, you carry it up? I think amoeba is, if I'm not mistaken, I might got this. I'm not sure. Just let me double check this. Stay with me before I say it's. Oh, okay, interesting. It's now stuck with me here. Let me quickly check this. Oh, okay. Cells in amoeba. My internet on the computer is not running well. Okay, like bacteria, amoebas have just one cell. Yes, as I mentioned earlier. Okay. It's unicellular organism, and so on and so forth. So it's one cell only. Okay, interesting. Okay, so why? Okay, now this is an interesting thing. You might think about it. Okay, you may think about it. Science is not done with these kind of questions. If you have something you want to share as a what this would mean for you, please share it with me again as a treasure hunt question. Let's see, what do you think about this? So some of the things they say sometimes, because many of the DNA could have repeated itself because of the environment and so on and so forth. It's 100 times larger doesn't mean it is more complex. Gene regulation in a human is still more complex. But again, all these are just hypotheses. Still more analysis is needed. So whole genome sequencing, it is a laboratory procedure to find the um, to find the nukes, the order of these nucleotides, to be able to read DNA. So start with DNA extraction, and then DNA fragmentation or shearing, library preparation, then to the sequencing machine, and eventually, finally, we have some computer program to run over this. Okay. Now, what you see here is whole genome sequencing using next generation sequencing, uh, which is through the the modern approaches. Okay, now we can achieve, looking